it seems like there's this risk that people learn something really useful from the evaluation, people, the tool developers, and then you would change the way you do the data analysis in response to that. I know when I saw my assembly software go into nucleotides and I saw all these misassemblies coming out, I said, oh, I know exactly why that's happening. And I went and I go fix that in the code. So then, of course, in a way, you run the risk of overfitting the software to the testing data sets that we have here. So how do we track the progression of this and sort of reduce the risk that the top scoring tool has gotten there just because it's been so finely tuned to do well on the evaluation metrics? Any thoughts on that?
So you could have some, I think you know the submission dates of software results.
Yeah.
So one can maybe restrict then results for tools to those on data sets after the submission of the tool. So basically, if the data set was available before, and then a developer submitted, so the gold standard for the data set was available before, and then if you have results for that tool on many data sets, you could restrict, like say you want informative evaluation only based on those data sets where the gold standard became available later.
Yeah, that's possible. That's actually what we did for KME1. So you will see what kind of results are were uploaded or submitted in the context of KME1 and what kind of results were submitted afterwards.
Okay. Yeah, if the distinction's clear, then you see what the most equal evaluation was when nobody had any ability to train on that data set, I guess. It could be really cool to see, how things are changing over time as well. I don't know how hard that is. But,
well, it would be possible, of course.
Yeah.
Okay, more comments? Phil.
This may be out of scope, but I wonder if there's also a component of, I attended a talk earlier this week on, it was totally unrelated to microbiome, but about FDA wanting to get away from clinical trials and go to more evaluating what was actually happening in the real world with these things, because the clinical trials were broken in a lot of ways. And so I wonder also if there might be a component, and then Alex and I were just talking about how after looking at the CAMI results, no one would ever use Ray, for example, as an assembler. But we've both had real-world experiences where Ray actually did quite well, and even better than some of the others on certain data sets. And so is there a component here maybe of collecting real-world data as well from people's assemblies on the assemblers they used and options and things, in addition, augmenting what we do in this sort of controlled setting as well, again, maybe to address that overfitting question.
Yeah, well, currently we don't have plans to do this. Right. We discussed this already. I mean, at some point going from like all these simulated data sets to real world data sets maybe. So, well, there are no concrete plans right now.
Yeah, just throwing it out there is something also to think about.
Hello. I also want to raise a potentially unpopular opinion, because I think people need to hear that, and you should consider maybe putting a disclaimer. So there are many algorithms out there that are doing very differently on different data sets. And there's no way to capture on your part with simulated data sets or selected some real world data sets to the entire diversity of data out there to make sure what's the top tool is going to be the top tool for every other data set.
Yeah.
I think binning algorithm is a great example, because all these algorithms are coming out with benchmarks compared to other algorithms, and they are being published because they are the best in a given data set compared to everything else. But when I take one data set and look what they are doing, there are myriad of mistakes they are doing, and we don't even go into really investigating them in these kinds of assessments, because we don't have the time or standard protocols to make sense of the quality of genomes, and we're missing a lot. So I'm very scared that this is a great, benchmarking is a very important aspect of how to make sense of where we're going. But also it creates a problem for biologists, because as computer scientists, we like to play with numbers, etc., but biology does not follow. The complexity of life does not follow our strategies to make sense of these algorithms and coming up with top tools that are doing so good. And I'm so scared that there's going to be a time a reviewer will say, well, you should have used this assembler for this data set because can we show that it's the best in the spinning algorithm, etc. Because these things change a lot, and by implementing standardized ways of benchmarking tools and creating lists, in fact, kills the creativity and the flexibility of microbiologists to really go into their data and use whatever seems to be the best for their data. So this, I think, is going to be a big issue going forward when CAMI continues to get better and better and bigger. So I feel like this creates a responsibility for people like you who are leading the field, and also maybe we need computer scientists to be a bit more insightful and careful about how to present these findings, because they don't really mean anything in the context of biology.
Well, I agree to a certain point. I agree, right? Because there are definitely assemblers that don't do a good job on metagenome data, right? Because they were never built for metagenomes, and people are still using these, right? I see papers where people are using Velvet for assembling a metagenome, and Velvet is clearly not a metagenome assembler, right? Okay, well, there might be cases, of course, and Phil just mentioned a case where, well, both of us had examples where Ray was actually doing a much better job compared to like Megahit or whatever, metagenome, whatever, other metagenome assembler. But there are also these assemblers which are very close to each other, right? And that's the same for binning tools. There are many binning tools which perform quite similar, actually. Right, and what people do is actually they run different binning tools, right? But how do you know which one is the right tool to use, right? And how do you know which result you should actually keep working on later on in your analysis? It's still a difficult choice, I think, right? So we need some sort of benchmarking at least to start somewhere, right? And also it's also meant for the developers, the tool developers, right? To figure out what are the real challenges right now. And where should the field go, and which are the challenges that we need to solve in the future, right?
I think, yeah.
Yeah.
No, it sounds like you're worried that the recommendations are too general and that they don't take into account the whole variety of data that's actually there. Mm-hmm. And there's I think this is something that's very true. There's so much diversity in the data that can be generated in the underlying systems, that what we've done in CAMI 1 is a starting point, but there's much to be done, and we should be looking at hundreds of different data sets of all kinds of types. And this is where I would hope that this could go at some point, that we have large collections of tools and large collections of data sets, and we can run these automatically and then figure out, I don't know, for data sets with this organismal complexity, or communities with this organismal complexity, or this abundance distributions, these kind of tools are good. So what CAMI 1 has, the results, it's a starting point, but it's by no means the end. And I also don't think that there will be one tool that's best for every setting or for every question. We already saw that in CAMI 1, that different programs have different strengths, and it can also just highlight what's good for what. So it's just, helping to provide guidelines, but not simplify too much. I agree with that.
I'll add, I'm not sure. I've got a different perspective on this, because I feel like the biology should be what's driving us. And the problem that we have now is that if you have a biological question that you're interested in, it's really hard, generally, for a biologist especially, to define some kind of metric under which they can work out how well an experimental approach is going to allow them to answer that question. So if you care about marker genes, we had somebody a few minutes ago interested in those, and you want to know what the diversity of this particular set of marker genes are. As an example, depending on which assembler you choose, the assembler might co-assemble everything in the marker genes from among a bunch of strains, or some assemblers are very careful and they don't pop bubbles, and you get all that diversity. And so I think beauty is going to be in the eye of the beholder here, and people need to be empowered to be able to create the metric that answers the question. So I would love to see CAMI provide an avenue where, for a tiny amount of work writing code, you could define the thing that you care about and see how all the different software stacks perform with this.
Daniel?
So Alice, yesterday you said this is in the, there's been critical assessments in the past, say for protein folding. And the nice thing about protein folding is that you can actually do the experiment and figure out what the fold is. So there is ground truth. And I guess that's the problem with any kind of microbiome evaluation, that we don't know what microbiomes look like. We don't know how many phages there are, what's going on with the plasmids and stuff like that. There's so much we don't know that any kind of evaluation is going to make lots of assumptions that people are not going to agree on. So that, I guess that's the problem that was mentioned previously, that methods are going to do differently. CAMI might make them look crappy, but that's because the model that CAMI is using for microbiome doesn't fit reality. So that's I guess the main problem.
Yeah, I think that's probably very true. I mean, how diverse, how many strains are there in any particular microbial community right now? Nobody can really tell you, because the only measurement level we have is OTUs, and you don't know how many strains are hidden in one OTU, and how much you cannot pick up from the lower abundant strains that you might never see if you do shotgun sequencing. But wouldn't you agree that you could kind of sample the range of possible parameters or of values, like regarding organismal complexities or possible underlying distributions, and then see which tools perform well across a wider range of these, so these kind of things. It would be lots of work, and until the point where we can have better estimates of some of these important properties of communities.
I think if you could do a really good simulation of microbiome data set, then you know so much. I mean, it would be great if we were in the situation where we could actually simulate microbiomes. That would mean that we understand a ton of stuff that nobody understands at the moment. So we wouldn't have to do any more analysis, because we basically have such an understanding of what microbiomes look like that
I think you've highlighted that we have a huge amount of work to do with simulation. So,
yeah.
But it's good, we know our models are wrong, and we just have to keep updating and refining them until they fit the data better and better. But they're still going to be wrong. They're always going to be wrong. We're never going to have perfect simulations for any community, I think.
Yeah, I mean, that's true for every field of biology, is it? Also evolutionary simulations, things like that.
Yeah, but the protein folding, you can actually
fold
the protein and see what it looks like. You can. In most cases, you can. That's why we had so little number of pictures. It's very hard. It's a very hard problem. I just don't want people to think that we can tie those folding predictions with ground truth all the time. We have a very small number of proteins we in fact know how they are folding, and they also change depending on the environment.
Okay. So are there more comments? Otherwise, I would just show you if you're interested, show you the where you get the data and where you upload the data, just to get an impression how this would work. So this is the central web page where we provide the data sets, right? So if you're interested in taking part in the next challenge or just using the data sets of the first challenge, they are all available here on this website. So you go to up and download, and this is where we make the data sets available, right? So this is, these are the new toy data sets for the second challenge. We always provide these toy data sets so that people can get familiar with like file formats and things like that. Yeah. So the challenge itself will look very similar in terms of the data we provide, except of course for the results, because these data sets have all the ground truth already. So you can download the data sets and you can do your evaluation right away using these toy data sets. For the challenge data sets, there are only reads available, right? And you have to do all the analysis. There are also as I said, the old data sets are also available. So if you go to the second page, you will see the data sets of the first CAMI challenge, and you can download it. So the toy data sets we provide right now are special environments this time. So it's not just a random collection of genes or genomes, as in the first challenge, but this is simulating a human microbiome in this case. And maybe I can increase the font a little bit. So in this case, we have 49 samples from different body sites, basically. In total, we have 245 gigabases of data. And we do have Illumina reads, 2 times 150 base pairs. And we also provide PacBio, simulated PacBio reads, which have an average read length of 3 KB. That's another 245 gigabases of data. And so everything is basically in one OpenStack object storage bucket, which you can just download by a single command. What you need for downloading it, because it's an object storage, it's not an FTP website. You need this client. So you have to download this client. It's just a jar file. You download the client, and then there are examples down here how you can actually get at the different data sets. Right? So the easiest thing to do is just to list all files with this command here. And depending on the data set you want to work on, you have to put in a different URL here. So for the different sampling sites, there are different URLs you could use. So if you want to look at the airways samples, you just copy paste this. Let me show you how this looks like. Just copy this URL, and then I downloaded the client already, the Java tool, and you would just say, okay, list this with the URL, and you get a list of all the files which are available. And these are quite a lot of files, because we have these 49 samples, and for each sample there are probably, I don't know, 10 files or so, right? We also provide the gold standard assembly again. The gold standard, as I showed yesterday, the gold standard is everything which is basically covered by at least 1x coverage after simulating the reads, right? So well, first thing we do, we simulate the community, the abundance of the different genomes, and then based on the abundance, we simulate the reads. And then we know where all the reads come from, which position in the genome, and we just look at this position in the genome. Whenever you have at least 1x coverage, this is the gold standard assembly. This is made available as FASTA files, and all the reads are also available as FASTQ files. And we also have this hybrid kind of gold standard assembly, which is a combination of the long read and the short read data, right? So if you have a perfect assembler, which combines both data sets, like types of data, let's say, you should evaluate this on the hybrid data set here, on the hybrid gold standard. So this is an example for the Webpack Biodata. The reads are provided as BAM files. So you see all the BAM files corresponding to the different OTUs. So each OTU is one genome in this case. Or maybe even several genomes within the OTU. But anyway, so the OTU number, basically, this is one genome you want to look at, and the BAM file has all the sequences from this particular genome. And as you can see, there are lots of files because there are hundreds of genomes in there. So you have the Webpack Biodata. You get, well, you not only have the BAM files, you also get the FASTQ files somewhere. It's a pretty long list. And that's the same for Illumina data and so on. If you're only interested in parts of the data, you can use regular expressions, basically, to download only parts of the data. So it's the data sets are described here on the web page. What's basically the content of each file. And if I want to download any of these files, I just use a regular expression together with the KME client. So if I want to download FASTQ files, for instance, I just use this expression here, fq.gc, and it will download everything that matches this regular expression fq.gc. So this is how you can actually download parts of the data if you don't want to download like 500 gigabytes of data. Okay, so that's the first data set, and the second data set is a Mouthguard data set that has similar size, so it's even larger. So that one has 64 samples, 320 gigabytes for Illumina data, and another 320 gigabytes for Webpack Biodata. And again, you can just download this and work on this. And the idea is, or for those of you who haven't seen this in the first challenge, when you run your tool, you have to upload the results, right? And the way it works is that you are, again, using the client for uploading. And we didn't want to make, well, let's say, so we had a deadline for uploading, right? Or at least submitting your results. But we didn't want to have that our servers get like overwhelmed because everybody is trying to upload this in the last minute and uploading like large data sets. So the way it works actually is when you run the Kami client on your results, you get a hash basically of your file. And what you do is you just upload the hash before the deadline, right? And then you have a couple of days to actually upload the data. That doesn't really matter because if you change the file, the hash will change, right? And we know that you were working on the file for another 2 weeks or whatever. So there's no pressure to upload everything in the last minute. You will just get some sort of identifier, register your data set with this identifier, and then you can upload your files again. Right. Oh, okay, and everything is actually anonymized. When you upload the file, there's basically only one person who knows who is actually uploading which file. So you get an anonymous ID, and that's something that came up yesterday the day before yesterday in the other meetings, in the CAST meeting, for instance. They do this assessment anonymously, right? And that's the same. We also do this anonymously. So when we look at the results the first time, we don't know which tool is actually which, right? We have these anonymous IDs, and we try to be like very, well, let's say, objective on the results and run the evaluation, and nobody actually knows. And what we actually did, we had a workshop with all the developers, so we invited all the developers so that they can actually look at results without knowing which tool is actually which. They might, of course, recognize their own tool, but they have no idea what the other tools is, right? And this is how the evaluation works, and then at the very end, we reveal the names of the real tools. Okay, questions regarding the data sets?
Yeah, something nice that was also mentioned yesterday by the CAST people was that they had external assessors invited to interpret the results. The assessors also got the tool results anonymized, because apparently when the assessors knew which tool is which, they wanted to change the rankings. So you have this in your head, you have this predefined notion, oh, this tool is good, and then, oh no, it can't be that bad, maybe it's that. So I was wondering if that's something we would want to have in Kami. And they were very positive about this concept. They said also the assessors then provided good feedback on which challenges are the most relevant and how challenges should be adapted and so on. And then the question is, who would be good assessors? I don't know if it would be tool developers or users of tools. Does anybody have ideas on that? Or potential suggestions who might be good assessors we could contact?
Volunteers?
Yeah.
May I ask a question?
Yeah.
Yes.
Well, last time, Kami 1, there is a gap of time between the assembly and the binding tool evaluation. So, will this be the same this time, or there will be out simultaneously?
It
No,
will
it's the same, right?
Yes. So so that there was this gap, so that for the assembly challenge we didn't want to provide the assembly gold standard, because else this challenge would be kind of, the solution would be known, right? And then so it should be the same way this time, that then after the assembly people have to hurry up, so to say. They are the first. And then afterwards, spinners and profilers that take assemblies as input, then they will get the perfect assembly.
Yeah. Because if the bidding tools use different inputs, it's really hard to compare, right?
Yeah.
That's why we split it into basically two challenges.
Yeah. One more. So, so the timeline. Sorry, I'm not sure if I missed this part, but so what is the timeline right now for Kami 2? So like the time, well, I guess there's still several months for the assembly tool submission, and there's probably another 3 months or 4 months for the bidding tool submission, right? So do you have the exact date for that?
So at the moment, we're still preparing the data, and because it's large data sets, this takes quite some time. That's a bit of the bottleneck. But so we hope to be able, so we might release challenges not all at the same time. So we'll be releasing, like starting several challenges, and we will start them as soon as the data set is ready. And one thing we learned also from last time was that people would have liked to have a bit more time. So we'll try to have the challenges run for 6 months each. And we might be starting with a pathogen detection challenge, which is from clinical diagnostics. So it's this is real metagenome data, and it's real. The data comes from a real situation in the clinic where it was important to diagnose the pathogen to decide on treatment, and this will be the challenge for people to solve, to find out, to predict which pathogen is in the sample and is causing the patient to be ill. And then, did we already talk about the challenge data that we will be offering? No. Okay. And then, so other data that we will be providing a marine data set. And there will be the same tasks as in the last challenge, so profiling, binning, and assembly. And then a rhizosphere data set, also with these tasks. And then finally a strain madness data set with lots and lots of related strains. So whoever wants to really know they're doing well on resolving strains, this data set will be the perfect thing to tackle. Yeah.
So if you, there's actually a mailing list. You can register on the mailing list if you're interested, and then we will inform you once the data will be available. Or follow us on Twitter. That also should work.
Wait, one more thing. Well, just yesterday, I received an email from the Mosaic team. I'm not sure if you have heard of that. The one by Janssen Microbiome sort of thing. So they're also having challenges. So I wonder if you are going to, well, these will be 2 independent challenges, or you guys really have some connection between each other? The Janssen.
Yeah, I think right now it's a.
Have you heard of that?
Yeah, I think right now we are separate. We are,
okay.
Yeah,
okay.
we talk to each other. So we see, maybe at some point we join forces, but right now it's separate.
Got it. Thank you very much.
Yeah.
Yeah. So also we have here a team of Philip Morris. We have a representative sitting here they have started a challenge and a profiling challenge, which they are evaluating at the moment. And so we offer to other challenges also the possibility to provide the data in our benchmarking system. So that's the plan, that there will be like one resource for all of it. Or if other teams like, they could, once the system is ready, use the framework for running their challenge. This would be our aim at some point.
Yeah. Yeah.
I think this might be relevant just in the context of getting involved. Just sort of a plug, but really a service, I hope. So last time we provided through, so I work for the Pittsburgh Supercomputing Center, and part of the Exceed project that is the program in the U.S. to provide computational resources to researchers. So we made these freely available for the last CAMI challenge for people who needed larger resources or bigger memory or whatever. So we're planning to do the same here. So hopefully that's an equalizer as well, that people's results aren't based on your access to computational resources. So there will be more information about that as well.
Mm-hmm. Yeah, thanks.
Do we have any, this is also an opportunity to shape future challenges. So the things that we're doing in this challenge, we got feedback at last year's ISMB. So if you have a wish list of what you would like us to tackle, feel free to let us know, and maybe we can make it happen then in the 3rd CAMI challenge. So something that was coming up last year also that we couldn't do this year was a meta-transcriptome challenge. So this would be on the to-do list, but maybe there's more things that you would like, that you would be interested in.
Daniel.
I know it's difficult, but functional binning or profiling or classification.
What would be your idea of evaluating performance?
Oh, what we're doing at the moment, we just, we take reference genomes and look at the genes that have been annotated and then try and recover those. I mean, that's very simplistic, but along those lines.
So functional bins, yeah. So this could be a quality metric we can add to the binning performance evaluation. If a method, it doesn't have to get the genome completely, but maybe the functions in the bin.
Well, the method might not be doing taxonomy, might just be doing only
Just function.
Yeah, and I mentioned that briefly yesterday. We've done a lot of evaluations like that where you're either looking just at the gene functional profile. You can either stratify that by which taxa are contributing the genes or not, and just look at the functional profile independently of the bugs. Or you can try to put those back into pathways and do different variations on metabolic reconstruction, either getting your gold standard from the original genome annotations or from a perfect assembly, depending on how hard you want to make it for the profiler. So there's different, I'd be very interested in something like that too.
Okay, I think we could evaluate that on the current data sets.
Yeah, I guess that was my question. Is this something that we could do without changing the simulator? Because then it's as simple as writing another evaluation metric.
Yeah.
So what we found is that it depends on whose annotations you believe. If you want to take the annotations directly from the underlying genomes, you can just directly use whatever you're dumping into the simulator. I've found it to be a little more interpretable to re-annotate everything uniformly somehow. Either re-annotate the incoming genomes or use something like a perfect assembly and use that as the annotation gold standard. But it's either way, it's still pretty close.
Thanks, Curtis. I think probably hopefully people will continue to have ideas about what we could evaluate, and it'd be nice to know just what the right forum is for discussing these ideas. Ideally, as a group, is the mailing list the right place to do that, or do we need some type of discussion forum? Or maybe we already have it, and I don't know.
Yeah, so the mailing list is if you have concrete questions. We try to organize meetings. So, I mean, there's this meeting. We will probably have another meeting in Europe at some point for people who want to discuss more intensely in person.
Yeah, well, actually, there is this microbiome COSI discussion Google group, which is not very active.
Yeah.
So we could use this if there is like enough, I don't know, interest, let's say, in this.
I hate getting on airplanes personally, and so I probably won't see any of you for another 3 or 4 years. But I'm happy to contribute on a forum if it's easy.
Okay. Yeah, okay, so thank you very much for your feedback. So I think we take a 10-minute break, and then the speakers for the next session maybe come up if they haven't copied their slides on the presentation laptop, and then we start in 10 minutes with the next talks. Thank you.